Okay, so let's get it started. So this is week 7. We are discussing classification problems. It's already halfway through, so the total, we have 13 weeks, including your presentation week. Let me know whether you have general comments on the lectures so far, and so do we need to change anything, the style, or you feel you can learn more if I gave some of those concepts in a different way. Okay. As mentioned before, so classification is one of the most successful application area machine learning has been found. Over the years, I found if you are not doing single-cell data analysis alone after this lecture, you will get more out of this classification problem more in your real life compared to other lectures. For example, I have a student from a small animal clinic. Once they learned this and they start to build this classification applications to classify animals into different categories according to the profile of all these clinical measurements, see whether this cat should belong to this group of disease, and they build those around. So today we just will go through this most classification approach commonly used in machine learning. You will realize all you have already learned, those linear algebra, will be very useful in this case as well. And all the neural network, if you open the algorithm itself and boil it down at the end, it's all about matrices applications, multiplications. So because you know how to do the matrix multiplication and you know how using adding machine, if you only know how to do the addition, you know how to do the multiplication, you know how to do the matrix multiplication, because all the neural network at the end are the matrices multiplications, so you know how this adding machine is working. So everything is just by adding things up. So we will go through neural networks and KNN, nearest neighbor approach, logistic regression, LDA, naive Bayesian, and see how do this evaluation for the classification algorithm, the performance, and how you judge whether this classifier is good and another is bad, how the combinations of parameters will change the results. So we need some criteria to know how to make decision. And then we will take a look of some of these classification tools in the cell type identification. So this problem is not the best application, because in biology, we are not really rely on neural network training to classify your cell type. Remember last week we have a computer session, and I let this Dr. Salinas to represent how he did this annotation manually. So that process will not be replaced by a neural network or classification algorithm itself. Not so easy at this stage. Maybe several years later, this AI can replace human to do this. But for now, this application classification tools don't have a lot direct applications in the cell type identification or other single cell data analysis. process. But if you can find the right problem, then you know all this background and how the algorithm is implement the principle behind those classification algorithms, then you may find some application areas in single cell data analysis. As I said, it's very important topic, not by single cell data analysis, but by just general machine learning field. So we know there are supervised, unsupervised algorithms, clustering problem are unsupervised, so you don't need training data. The algorithm will automatically decide the membership of this data points belongs to which clusters. Well, classification problem, we normally need a training to have some of this ground truth knowledge about the separation of the data points or samples that allow this training data to train the classifiers, okay? Then you get this trained model or set up, or it could be a combination of parameters. Then you have the classifiers. Then you can apply your classifiers to the unknown data, so the new data, and then to get the separation to which cluster, which classification group it belongs to. So this is called a supervised learning. So that's a two steps. First, the learning step, that's where you use training data to train the model, to train the classifiers, and to get it set up. And then to do the real classification steps. So in the first step, the model is constructed. You have to select which model, which algorithm you use, and then train the model to set up preset classifier. Then the classification steps, you use construct the model to label those given data, okay? So classifiers refers to machine learning functions. So there are many different forms of what kind of classifier it looks like. It's implemented by all different kinds of algorithms. Okay, so classifiers define a very general machine learning framework. So you have labeled training data. Then you may want to have some feature selection or extraction of features, or QC. So those are the basic steps. You want to enhance the signal or reduce the noise in the data. Next week, we will focus on some of these feature extraction, feature selection steps. But now we just skip, okay, directly goes into the machine learning algorithm. In this case, is a classification algorithm training. You will transform, or sometimes you do the convolution step. I will show you how to do it. And you basically will give the algorithm the vectors. Okay, vectors are the data points. This thing will be vectorized. Okay, in single cell data, it's very easy because you can just get every individual cell's expression profile. That's by itself vectors. But if you give an image, that's a 2D, is not a vector. If color image, you have a multiple matrices combined to give you color. So you need to have a way to vectorize your data. Anyway, for the current framework, the vectors are the data, okay? You gave to the machine algorithms. So once this has been trained, then you model, your classifier is done. So you can go to the deployment stage, start to utilize this into the real data, and then you put the input data, then let this classifier to separate them into different clusters, different classes. As I said, this classifier can be in a different form. So in mathematical way to express it, you can see, okay, this is a function. The function is your classifier. So this is a very abstract representation. You have a function, you take the input. So input here as an example is a picture. We have to find a way to vectorize it, easy to get into the function, right? Then you output of this function are the classes. Apple, tomato, cows. Those are IDs, labels. So that's a very generalized representation of classification algorithm. You think about the function of F. As I said, function can be many different forms. Okay, it can be a neural network, can be a logistic regression function, can be a LDA, a procedure. So we will learn this different type of functions to help you to do this classification. So that's in the function format to visualize the training step as well as this testing steps. So this function is also called a prediction function, all right? So you need to have a certain number of this training, okay? Not only a few, you need a large number of training data in order to fine-tune the function parameters to give you the constructed model. So this model will be utilized as a function to take input the picture, and the same kind of input we put into the training. And then the prediction will be the labels of this ID of the classes of this input data. So any questions for this very general description? Okay. By the way, in the single cell world, because we don't have a very good training data in general, so this procedure will have some problem because other people's trained data, they may have different features which is different from what your data is. So you may not be able to get the best results. Maybe you have some examples in the applications. Some very so-called fundamental models have been trained with a very large amount of this data set in single cell world, but in real applications, we don't find very good results compared to the average marker gene based annotation. I will show you some example in the computer sessions Thursday. Okay, so in general, I believe single cell, we don't have this good high quality training data, and because we also don't have a ground truth, some cell type is in the middle of a mixture of a stage, so you don't know this feature belongs to definitely this cluster of this group of cell or that group. So we don't have a clear high quality training data, not like this picture, it's a dog, it's a dog, it's a cat, it's a cat, nobody will be confused about it. Okay, this is a general problem. So those are the classifiers we can find in the Wikipedia, for example. There's highlighted ones. Those we will mention through the class, logistic regression, Bayes cluster. I will show you the perceptrons, and this is neurons in the neural network, how it works. Next week, we will talk about the support vector machines, so that we will introduce your kernel or the concept, decision trees next week, okay? Neural networks. Okay, so this is a perceptron. So this is the most important topic I'm going to discuss today. This is in the early stage, data scientists and computer scientists try to simulate the neurons, how a neuron works. So neuron have this neuron body itself, then there what's this called? Axon? Yeah, so some input signal will be integrated, and then the state of the neuron changed, then gave you other different kind of electric potential and pass on the information to the next neurons. Okay, now let's just focus on one single neuron in this literature in the computer science is called a perceptron. Okay, perceptron is it's a small device computationally. It's binary classifiers, and you can use this as the most fundamental unit in the neural network. Okay. So this is it contains this multiple weight matrix. Not a matrix, it's a weight will be a vector. So you have a vector contains n numbers of these elements. So this is to tune the input signals, and then you will have a summation operation to add these weighted signals together, and then you decide whether based on the summation of your total, you will decide whether this perception will be active or inactive. So your output could be 0, 1s, or if you want to use different schema, then you can have a positive 1, negative 1. Okay, so the output will be just binary signals. Okay, so this is from the very simple basic unit of all the neural network you are working with. So I'm going to give you a real numeric examples to see, okay, how we can do this. So this is my perception here. I have 1, 2, 3 training data input. Let's see. So if my, let's use this. So I know the answer. So if I have input data, if I, for example, use this one. If I have input data 1 minus 2, 0, 1. So that's my first input data. You know you can vectorize your input data into a vector. So this is a vertical vector. Then let's just set my perceptron, and this have a weight matrix. So this is a 4 dimension, 4 elements in the input data. So each I'm going to set a weight. So if I set weight as a first set, this 1 minus 1, 0, 0. Okay. And I also know the answer for the output 1 should be output one, I know the answer because I'm training this model. I know the answer is this negative one. Okay. So how can I integrate the information and to get the output? Very simple. I will just times this times this times this, then add them together. And then I will see the sign function of this. If this is a good classifier, I should be able to predict this results as a -1. So I can try multiplying. This is 1. This will be 2, 0, 0. Okay, 1, 2, 0, 0. And sum together, my number, the result is 3. Okay, so for the first one, I have a 3, and the sign will be +1. So it looks like this is not a really good perception for my input data because it gave me the wrong answer. So what should I do? I will change the weight. So this matrix weight vector. So the weight vector is gave you the property of this particular perceptron. So if you realize if I change this to the second set, if I change these parameters, -0.7, 1.4, 27. Okay, I'm going to do the dot product between this. Okay. In MATLAB, if you can do it, then you get the correct answer. Okay. So this is just one input. I also need to train the data. For the second input, for example, input 2. My input 2 is 0, 1.5. In my example, -0.5, -1. The output I expect to have is output. I expect to have, again, -1. So you can test. If I don't change the parameter, I use the first set. It not give me the right answer, but this particular one gave me the right answer, -1. I can further try my third input. This example is 0.5. Okay, output. So for this three training data, 3 data points, this particular combinations of my weight is a solution will satisfy all of them to be predicted correctly. then this classifier is set. It's already been trained. So if I have another unknown, if I have another input, say orange, input 4, I have numbers, okay? 0, 1. So I have numbers that will give a prediction. So I use 3 as a training, okay? 3 training data. I use 3 training data to fine-tune the parameters of my perception and test with 1 another input data. Okay. So this training data can be really long, can be a huge matrix. You will realize in order to train this, you can do this matrix multiplied by this vector, and you will get the product, dot product between each of these training data with this weight matrix. And then you take the sign function. If the positive gave 1, if the negative gave -1, so you have output. So this is very easy to compute. Okay, so over the matrix input dot product with the weight you set up. So that's what so that's the input. So you see these 3 vectors, okay? And this is 2 sets of this weight combination. If you do dot product, this will test both sets of the combinations, give you this result, and then the sum. And you realize the second, this set gave you the right answer, that's what you want, -1, -1, 1. Well, the first one gave you the wrong answer. If you randomly put this, it may not give you the best answer, so you have to find a way. This is a small example. We can set this value slightly differently, only care about the signs. Okay. So determine the weight. So you can optimize the combination of those numbers. And you may realize this sum function here seems lost a lot of signals from the aggregating information. So sometimes you want to replace this with other activation function. You may say, okay, I don't want to be binarized only by the sign of the summation of signals. I want to have a sigmoid, and this gave me output based on my input value between -1 and 1, and this they're transformed into 1 to 0 based on that curve. You can have other, so you need to be positive, but after you become positive, you have this slope rather than just binary. Okay. So this function you can next follow the next right after you summarize the signals. Then you can do the transformation. Basically, give you the output in a different way. This is one of the activation functions to transform your input values into value between 0 and 1 follow this particular function. So based on your the type of your activation function, you can say, okay, my classifier is linear or linear regression based with the sign. This is a logistic regression based, okay, using this sigmoid function as output. Give you a slightly different flavors of this. You have a hard decision, and yes, no, and then this gave you a soft decision, okay, based on how extreme your value, and this just gave you some kind of regression signal. So that's examples. Okay, in our example, we have W have 4 numbers, we have 3 numbers, then use this, and you can classify data into 2 categories. Okay. So if you open the source code for, this is Python, this is MATLAB, you start to see the perceptron definition. You have training, input, output, and inside here you have this dot product between your weight vector with the training data, okay? And then here using a sign function to see whether this is same to the ground truth. Why? Because it's training data, you know the yes or no. And if it's good, then you get this small values. You don't change your weight. If it's very different, because you predicted wrong, then this R will have a larger values, and you change this correspondingly. After several loops, then your solution will be, your W will not be changed further, so you stop the training, okay? Okay, here is things to get more interested in, okay? Now, you know this multiple inputs and information will be aggregated into one perceptron. So that's what you'll see from here. You have a multiple ABC inputs, and then the information will be aggregated and put into one perceptron here, right? Well, the neural network idea is you put multiple perceptrons together side by side. So this would form a one layer. So your algorithm input will not be only give to one perceptron to decide to active your activation function, but you also will give information to a different perceptron, okay, multiple simultaneously, and each have its own weight, will be trained. And also the activation function can be different. So this forms a network. So one set of signal give to multiple perceptrons. Let those perceptrons to aggregate information and activate this function and give output. And those output will be further passed on to another layers. If you consider these layers as a previous layers of a perceptron, then it will pass on the information to the next layer. It can be increased number of the neurons in that layer, or you can decrease. So you can have options to how to expand your information and weight differently, how to compress your information with fewer neurons across different layers. But this is a concept for putting perceptrons together to form a network. This is a multi-layered networks. Well, in order to do this, at the end, you will have this matrix multiplication operation, because you know the input will be multiplied by dot product with your weight. Okay, that's inside. But when you have multiple perceptrons, you have this weight vector become a matrix. And you also have a previous input, or the collection of vectors is also a matrix. So at the end, you are doing matrix to matrix multiplication. Depends on your dimension, okay? So if you only use a single layer, then that's inputted in the square, this box here. There are input signals you measured or from a data point itself. Then you have one layer of this perceptron and then give the decision, the output. But if you decide to make multiple layers, and then it becomes multiple layer neural network. If the layer, the number of layers increased, then the neural network become a deep neural network. So your input layer and to the output layer in between may have, say, 5, 10, or even more layers. So that's the concept, so-called deep neural network, but the foundation is the perception, okay? So it depends on how you set it up. So you can have input, have a lot of perceptions to accept the information, especially if you have a large, high-dimensional data. So you need a very large number of those perceptions to aggregate, to synthesize those information. But in the middle, you may have very narrow bottleneck neuron perception layers. And then output could be expand again. So this creates so-called autoencoder structure. This framework will allow you to encode those high-dimensional information into very low-dimensional internal layer perception layers. And then you can reconstruct those information which you get from input through this bottleneck, expand their weights and into the output, which restore the dimensionality as your input data. So this autoencoder will learn the input and the output, and through the training data, it try to simulate. The output will be compared with your input until they become very similar to each other. You cannot distinguish them statistically. So after that, you finish the training of your autoencoder. So this is a very special framework in order to do this information compression and then expansion. And the training data will be by itself, okay? Try to learn itself. Why we are doing this? Because we try to learn the features inside this hidden layer, because they are low dimensional, so the information will be compressed and captured in this intermediate level of low dimensional space, okay? So basically you are extracting information from a high dimensional space data and summarize their most featured information into this intermediate level with a few neurons, low dimensional space representation. And then you can use the information from the hidden layer, so inside here, to do the generative model. So you want to simulate some data out of this autoencoder framework to see, okay, I want to generate some data looks very similar as input ones. Okay, you can do this using low dimensional information inside this intermediate level, middle level layer of this perception. Okay, so as I said in the outline today, so we are going to talk about applications of classifiers in single cell type annotation. And here I want to jump directly to this application because it is relevant, and this is a so-called foundation model for single cell data cell type annotation. This is published last year in a Nature paper, which they created a tool called assimilarity. Okay, that's for single cell data type annotation. They train their neural network using a large scale, 23 million cells from Cell Atlas projects. Well, the cell has been annotated using traditional method. Okay. Well, they cover a lot of cell types, and then each cell type they have a lot of cells for training. So they train their neural network using this method in order to get this large different type, maybe hundreds of the cell types, classes, and their features to build into this huge neural network framework. So that's where the tissue and the samples and their from 56 training studies and test or label studies. So they aggregate all this previous published data and together to have a large training data set from multiple tissues, cell types. They also create this cell ontology. So T cell will have a CD8, CD4 subclusters. Well, CD4 can be further classified as a regulatory T cells. So they have this not only the cell type itself, but also the relationship between cell types. Then they have some special setup for training, because in general we have this training data into their known class, but they set up these 3 cells, triplets, and with the 2 cells, one is used as an anchor, another is positive, and you know the answer, and then another is negative. So instead of one trained the neural network with individual cells, they use combinations of 3 of them, one with a very high confidence, the membership, their anchor cells, and then another is positive, another negative, as a triplets to train the neural network. So instead of giving you yes or no, which cluster, they have 3 cells each time to have this triplet sample. You need to be all right, and the 3 to be correct, predict this 2, and predict this as a negative in order to give the correct answer, right? So they train this and then get the positions at the end of all this trained data. At the end, that will be a vector database. Basically, you have point to all the cell types into this hyperdimension space. I don't remember the number of the parameter each, maybe 128, some combinations of numbers. Then they have a dictionary, say, okay, T cell will be that position in this 128 dimension space. And the CD T cell will be same direction, but slightly toward that direction. CD8 will be there. So they use this vector database to as a predictor later. So once you train this finished with all these several millions of data in different four tissues, cell types, then you get this vector database, okay? So this only issue use 2D, but it's high dimensional. And once you have your query cell, so in the annotation, every cell will goes into the search, and this cell will have its profile of expression, and it goes through the neural network to project, basically embed this into the 128 dimension. And then you will search the database, say, okay, your new cell will go to that direction. So it's not a T cell. If another new cell comes in that goes to that direction, or in our database we have the T cell in that direction, then that new cells will be assigned as a T cell. So the distance they use is a cosine distance that just point you to that direction in the dictionary of the cell type. Okay, so if you look at the description of their method, they designed this distance, embedded the cell into the low dimensional space. We can check how low it is. So your input will be thousands of genes expression, but if you have 128 representation, that's relatively low dimensional space. And then they use triplet, this special setup as a training to calculate the good or bad the prediction is. So loss function have this triplet definition. And they use cosine distance to test your input data to point you to that direction. So to assign each individual cells a number class. Okay, so so this is we will try this in Thursday and then see whether we can download that vector database. It's a huge file, but then you can test each of your cells against this atlas dictionary and assign a cell type. So before the break, I want to introduce this convolutional neural network, CNN. It has been used in this picture or image classification problems. So this is like 2D data, okay? So you have X, Y position of each pixel and also their colors, so they have multiple layers of this 2D matrix input. So we need to find a way to vectorize this to give to neural network, right? So how we aggregate this information to vectorize these pictures, the procedure we use is called convolution, okay? So you will see it will have a scanner, okay? A sub matrix will like a sliding window to scan your picture from left to right and the next row, and the information will be summed together and into another layer and then scan again. So through this aggregation procedure, your input data will be embedded into a vector. Okay, this vector will give to neural network to predict. So this feature of this pictures will be, information will be aggregated through this convolution procedure. So I'm going to play this, and then you take a break. Okay, so how the convolution procedure will work. Low the volume.
And Doug.
Thanks for coming in to talk about Doug Jr.
Of course.
Okay, so this picture is your in your left. This picture is A. Okay, the scan. And the values of this small window will be added together and put into another layer. Yeah, so after this multi-dimensional scan different ways, and then you summarize your extract information to a low-dimensional feature. So this data will compare with your trained results, and then it's 92% match with A, so it will be assigned to the highest probability category. Okay, so it belongs to A, which have a very low probability belongs to C, okay, instead. So that's a way to do this, convert these 2D images by scanning and learn the sub-features in this 2D dimension matrices, and then to learn the feature, to build a feature matrix. Okay. Okay, let's have a break. Come back in 5 minutes. Okay, so for the convolutionary operation, you may have different ways to, for example, take care of whether you want to take care of this boundary effects, okay? The different ways to do it. But anyway, so we need to understand, so now, for now is what is machine learning, what is deep learning. Deep learning is a very specific word for machine learning method based on the deep neural network. So you know what a deep neural network right now is, but most of this is for classification problem. Okay, so KNN, so this stands for, so that's we switch, move to next method. So KNN is the so-called K nearest neighbor. This method is a very lazy method. If you decide I want to look at 3 neighbors in order to assign my membership, I will look at this input data and look at the neighbors, 3 nearest neighbors, and the 2 belongs to purple group. I will assign this star as a purple. If you decide to look at neighborhood with the members of k equals 6, then you look at the nearest neighbor 1, 2, 3, 4, 5, 6 of this particular given data. Then in this case, you have 4 members belong to yellow class and the 2 belongs to purple class. Then you assign this new data point into yellow group. Okay, so this is a very lazy approach, just by looking at its neighborhood, just by vote and see which member you belong to. So we just need to care about the distance, how you compute the distance between your data points with the training data points. You know the answers. Okay, sometimes you want to give greater weight for the near neighbors. If you have a neighbor which very, very close to you, you want to give it weight is 1 over the distance. If the small distance, this W will be large. So you gave those members which very close to your query point higher weight in order to compute the membership rather than treat every neighbors in that circle, in your case neighbors in your case circle equally, but you gave some fine tune about this distance and give greater weight to close neighbors. Okay, logistic regression is another approach. So you have your logic transformation and then do a basic regression on it to get the results. So depends on your calculation, and then this curve has been already trained and then gave you this particular parameters. You see this not in the 0 position or there, but this logistic regression curve will help you if you have a new samples comes in, then you belong to this category at a certain percentage, and this category certain percentage. So you can compute that directly. So this is through training, and you get the curve and then use this regression curve to do the classification. So I just use 1 page to explain this to cover so your terminologies. LDA stands for linear discriminant analysis. Well, it's pretty much like a PCA, but you want to find the particular rotation and then to separate 2 class rather than found the axis to visualize the maximize the dispersion. In this case, you need to consider the rotation matrix. You need to consider both group. So if you look at the red dots and the green dots, they are two different classes. Then you want to find a particular angle that separates them most, rather than treat them all as a single group. Like PCA will visualize the most different separation among the same group. You don't care, they belong to two or more different classes. So this is a particular visualization. So you want to look at your data through this particular angle. This may be the best separation because you have a centroid. This red group will collapse into your project into here with the center around here, which blue one will be here. The distance between these two groups are the largest, rather than treat them as one single group to be largest, that separation. So you need to consider two groups. And this will be not the best, and the distance between the two group center will be not the maximized. So this is a dimensionality reduction. So you reduce 2D into 1D. Depends on how you select your 1D axis. You try to maximize this mean difference. Okay, you have a green mu and the red mu. They're in the different location. You want to maximize their separation, and also you want to minimize within a group dispersion. Within a group, variation will be small. They are closely located around the mean, the best. And you want to minimize within a group variation, but maximize the difference between group mean. So you end up with this particular expression. So you want to maximize the whole expression. Maximize this means the difference between them will be maximized, and this within the group variation will be small. In this example, you will have big separation between red and the green group. However, this is not the best solution for LDA because the separation within the within the group member separation is still very large. There is some confusion in this overlapping area, which the best solution for LDA for this particular example will be you found the projection, let the samples to collapse into this direction onto this axis. Will they all separated and with a separatable difference between mean, and also minimized their variability within the group. So this is a very good illustration to help you to understand. LDA is trying to find the separation using the rotation matrix to rotate your data, allow them to project onto the low dimensional line or space. So they have a maximized distance between group mean and minimized within group variation. So that's an achievement for the LDA. Algorithm description for LDA algorithm. So you have input data will be your data matrix, and also with label. The label is vector y, and to give you the member ID for each of your input samples. And here we first compute the mean vector for each class. Okay, if you have 2 groups, the C will be 2. If you have 3 groups, the C will be 3. So each group you will compute the average. Then you will compute the total average mean vectors. And then within the class, scatter between class, there are several matrices you want to compute. Then there's a eigendecomposition procedure, and to do this, so you have this within the class scatter, and this 2 scatter measurement together, and then this is square matrix. Then you do this to find basically the direction of this vector. Okay. So this is real MATLAB code. So you have input X, the label. Okay, the vector contains a classification, and then see how many classes in your group. So we basically find out how many unique labels you have. Then this is a between and within class scatter matrix. So you compute the mean and compute these 2 matrices, okay, and then put them together to find eigen. And then this is solutions for your rotation matrix. Then you rotate your input X matrix. Then your projection Y is rotated. That will be a solution to separate these 2 groups. So this is a side-by-side comparison between PCA and LDA. PCA will not consider the labels, put everything together, try to maximize the separation. While the PCA, LDA will consider 2 groups. And for PCA, this is the best solution to this axis, because the separation among data points is maximized. Well, you need to consider the difference between them and also the within group separation. You want to minimize, not maximize the separation of the projection for within group members. So we are going to try this in computer session, okay? So if you get lost somehow, it's okay. We have real data and try to play with this. Okay, so naive Bayesian method, it just uses a Bayesian formula to compute a posterior probability by looking at prior distribution, probability, and the likelihood. So this is an example. So if you don't have any idea about which member of this new data points belong to yellow or green, so you compute the prior knowledge, prior probability, because you have in total 17 samples. There are 7 belongs to yellow, 10 belongs to green. So most likely, if you have a new introduced data point, it's likely to be green because the prior frequency are higher in green. So you're likely to be green. However, then you need to have consideration about the likelihood. So you consider its neighbors, then your likelihood to be green are 3 times higher than to be yellow. So you put that information in, and then you compute a posterior probability, and then at the end it's likely to be a green. Okay. So this is a direct applications for this Bayesian framework in a different
setup, you will consider, okay, those documentation, you should receive email, whether they are spam or not. Their probability to have this frame and then to be spam, then you compute so in here you compute, this is a prior probability, and this is a the likelihood. So you put them together and then you decide whether this particular phrase is belongs to spam email or not. So this is another user case to use Bayesian classifications. So in the single cell word, the one you click and then using marker genes as an annotation is some kind of idea is here. So you can consider each phrase here is a marker genes, whether this marker genes will help you to get this cell belongs to this cell type, the frequency, the probability will be used in the computation. Some genes, common markers, they appeared in many different cell types. So those markers will not have a very good, will not be weight very high in some cases. So those are the word frequency, the idea I just gave to you. So if you have a marker genes which highly frequent appeared in many different cell types, then they will be, that frequency will be taken into account, which will not be weighted higher in the decision of whether the cell belongs to which group. Other way around, if you have a marker genes which exclusively tell you, oh, this cell type is a neuron, so it's highly specific. So that kind of word will be utilized in a more weighted way. Okay, there are many readings you can have to expand if you want to go deeper to some of those particular areas. So I have some links for you to have a PDF. Then on the YouTube we have a lot of resources. So now we move to see how can we, if we set up a classifiers, so how can we decide to give a quality check. So this is a very good classifiers in overall in this type of a study or this type of applications. You need to have some idea what is cross-validation. You need to be able to explain, for example, what's ROC curve, okay? So in this classifier evaluation scenario, you always see CV, okay, you'll use several-fold of cross-validation to validate, okay, or the performance of your classifiers. So what does that mean? So if you see, okay, I want to do 10-fold cross-validation, 10-fold CV, then you will repeat testing your classifier performance, see, accuracy by 10 times, repeat itself randomly. Each time you will randomly set, see, 90%, see, certain percent, it could be 20 by 80, could be 10 by 90. So this, you separate your training data into the data that will be used as a training, and then use other part as a testing. So you know the answers of all your training data. You have labels. See, this is apple, this is a pear, that's dog. So you have real answers for those, but you separate your data into training one, small training part, and then testing part. Then you use training part to train your model, train your classifiers. Then use the testing part to test its accuracy, because you know the answer, so you can test the accuracy 93% in this round. You will repeat this procedure again. Randomly sample your training data, separate your training data into training part and the validation part. Then use the training part to train your model, run again, the parameter change. Then use the testing part to test the accuracy of your trained model. In this case, 90%. Then you repeat this 10 times. At the end, you will average your validation accuracy to give you the final accuracy. Then you claim, I test my method, my classifiers, using tenfold cross-validation, and I get the average final accuracy, see, 92%, something like that, because you average your 10 times. Okay? Understand the procedure? So you need a good training data. Okay, you have a lot, then separate them into validation set and training set. Okay, so we need to know how to calculate the accuracy. So accuracy is a true positive plus true negative. So the all correct prediction divided by the total everything. So all the case you have a true positive, true negative. That's a good prediction you make, and then you make some mistake, and this number false negative, false positive. False positive means they're not true, but your classifier gave it's true, but it's false positive, not true. False negative is in reality is true, but your classifier gave the answer as a false, which is not real. So you want to have a large number of this TP, true positive, true negative, with smaller number in this two position. False positive, false negative should be small in order to indicate a good classifier. This is called a confusion matrix. With this matrix for numbers, you can compute accuracy, you can compute recall, or false positive rate, where that's a true positive divided by true positive plus false negative. So this position, so you want to have this value big compared to the total. So that's a false positive rate. And you can compute sensitivity. That's a TP positive rate minus this is also sensitivity. Specificity, okay? There also this correlation coefficient. So based on the definition, and you need to understand the confusion matrix in order to know how this evaluation matrix will be computed. Any questions? Yeah, I got confused. Let's see. This should maybe something wrong. Maybe it should be 1 minus.
I think true negative.
Sensitivity will be if it's a sensitivity. If it's true. Accuracy, maybe something wrong, but I got confused. This is maybe wrong. False positive.
Specificity is true negative.
So it's definitely wrong because the formula are the same. so go back and let me know how to correct this. Okay, so if you haven't seen ROC curve and you don't understand this, so this is important. False positive rate is 1 minus the specificity, okay? So if you labeled your x-axis as a false positive rate, then this is from 0 to 1. Sometimes you'll see this label will be specificity. If you label your x-axis as a specificity, then you need to flip 0 once. The 1 will be here and 0 will be there in order to label this as a specificity. So in our case, this label as a false positive rate. So you want this false positive rate to be small. So you want to have your classifier have a small false positive rate. That means your point should be close to the y-axis. Okay, that's the indication of the good classifier. Well, at the same time, you want to have a high true positive rate. Okay, always recall sensitivity. So that means if you have samples are real, and you need to be calling that very in high frequency. So you need to have a good true positive rate to be higher. So you want to have classifiers in a different situation. Okay, so you test your classifiers in different combinations of parameter, for example, multiple times, and sometimes you get levels of this 0.2 false positive rate and positive rate, true positive rate 0.8. So this is a good indication. Then you try to build up this curve. A good classifier will have large area under the curve. So once you have the curve, you compute the area, this gray zone here. The larger the area, the better your classifier is, because your classifiers tend to have a small false positive rate and a higher, larger true positive rate. So that's a way to read this information. In reality, it's not so easy to compute this curve. So you need to consider different ways to do this. If you see this, read the paper carefully how this curve was constructed. Okay? But this curve is a very good indication of whether you have a good classifier or a bad one. So bad ones will confuse itself, give a lot of this false positive rate. That means this number will be large and your curve will be close to this diagonal position. This diagonal position will be definitely just random. If everything is random, so you get this curve located onto this diagonal straight line here. So if your curve pushed to the upper left upper corner, that's a good, because the area will be larger. Well, single cell classification problem, there are many studies that use different algorithms, like a single cell similarity using cosine similarity with the foundational model. Before even that, people use the cosine similarity to do this profiling classification, and then there are people use correlation and also distance to do this nearest neighbor approach. You can have a supervised learning. In this case, we need a training, so this use support vector machine we mentioned, and also Surette use random forest. Now, if you have a good training data, you probably can have a very good results. But the problem is the training data people use to build these tools will be different from the data you are going to use for the prediction. So you may have some bias in there. So we will try a different approach and see. At the end, you will realize marker genes may be still very good approach, and the manual annotation is necessary at this stage until later on maybe some smart agent can replace human, but not now. And all these different tools have been published, but I don't believe any of them are dominant the whole market right now. So some people use a single R. You can try, we can try and see in your real applications and see which ones give you the result you want. This is another list, and from a review paper. Most of them in the R and the Python. I don't know which one is really popular at this moment. So this is one of them, SC predictor. See where it is. It's here. So it's R. It's a SVM based with a video kernel. So that's training data in, and then you have PCA, okay, first. And then using the PC to learn those features which can separate your cells. It's already labeled, so you know the separation of the cell. And then use PCs to extract those PCs and use as a training to get the best training model. Then once you have the input, the data set you want to predict, then you have to do the projection using this PCA and load the features in and into this model to do the classification. Okay, so the idea is there, and not so sure here. So what's so they use SVM, which is a classifier method we will discuss next week. But that's the model they use after the extraction of the PCs, which give you the most separation of the two classes. There's another tool called a single cell net, SCN. That's a procedure, it's training. Also, you need looks like they have information from across species clusters, and build a classifiers using random forest. Okay, assemble this and start to do. So they use random forest. So output including this query theory mark. Yeah. So group your cells into different cell type. Some scores to each cluster, and this belongs to fibroblasts. So this one, it's pathway-based single-cell RNA classification, and also clustering. So it's also based on the random forest. So this study, which year? I want to find which year. This is an example. They have a T-cell subcluster annotation, and they use manual annotation approach by define these cell groups, gene programs using these marker genes. And then they just do this manual annotation without any classification algorithm. So this is the method we used in the toolbox. It's based on the PangalorDB. It was a web server which people can upload their data, and nowadays this service already discontinued, but the algorithm we implemented in the toolbox. This is the idea about this algorithm in their publications. So we need to compute a weight for each of the gene markers, and then take average for each clusters of the cell, use those marker genes information with their rate, normalized rate, and then we will compute each cluster group. Their score belongs to all different kinds of cell types. So in the database, we have about 140 different cell types. So for any given cluster, we will compute a score for each of these cell type. The highest score will indicate this group most likely to be that particular cell type. So that's what we did, follow this paper. I think that's all for this week, and we may spend some time on this algorithm in Thursday, and to see, open the M file and what's the implementation in there, and where the marker genes are defined, how the weight is computed. I think that's all for this week, and let me know if you have any questions. Let's see, I want to see what we will do a Thursday. Yeah, Thursday we will try perception and neural network framework, classified learning. That's all what we will do on Thursday. Okay, so that's all for this week lecture. Yeah. Yeah.
Question.
Yeah.
So when we work with the regenerative models or with injury models,